Hello and welcome to the Sheekey Science Show and the first ever Sheekey's Science Shoutout. That was quite hard to say. And so basically, I recently posted on the discussion part of my channel that I was thinking of starting a weekly science news update alongside my usual videos. And since no one told me it was going to be a bad idea, I thought I'd at least try it myself because I read a lot of content throughout the week and often I forget majority of what I read. And so at least by making these videos, I can at least make some kind of record about the latest and greatest science news. And when I say science, I'm probably going to bias it, as most of my videos, to biology and specifically looking at any new aging, cancer, neuroscience, biotech, CRISPR related science. So if that's all you're interested in, then hopefully you'll like this new series of videos. So without further ado, in this video we're going to look at a recent publication that showed the age accumulation of methylmalonic acid and its link with tumour progression. We're going to look at lipid-based nanoparticles that enable base editing in vivo, and lastly we'll look at some genetically modified flies to be released in Florida at the start of next year. So first up is this Nature article that came out just last week. Age-induced accumulation of methylmalonic acid promotes tumour progression. So the risk of cancer and tumour development increases with age, but the big question is why. And so one reason could be, as we age, we've accumulated more mutations that could put the cells at risk of becoming tumorigenic. But there's also other potential reasons that could explain this association. And so in this study, they showed that metabolic alterations that occur with age can produce a systemic environment that favours the progression and aggressiveness of tumours. So what they did in this study is they took human serum from either young or old healthy donors, and they gave this serum to different cell lines, different cancer cell lines, and studied the phenotype and genetic changes within these cells. And what they noticed was that the cells that were given the older human serum showed pro-metastatic properties. And so to understand why this was the case, they looked at the presence of different metabolites within both the young and old healthy donor human serum. And what they found was, as expected, they saw decreases in the levels of glutathione, spermidine, and glutamine, and alpha-ketoglutarate. But they also saw consistent increases in the serum of age donors, and the three metabolites that were consistently increased were phosphoenol pyruvate, which is a component of glycolysis, crinolinate, and methylmalonic acid. And so they tested each of these individually to see if any of them on their own could induce the pro-aggressive effects seen in these different cancer cells. And they found that the methylmalonic acid, MMA, could. And so what is MMA? So MMA is a byproduct of the metabolism of branched-chain amino acids, and it can be linked to coenzyme A, and that can be converted to succinyl-CoA, and then that can be fed into the Krebs cycle. However, this conversion requires an enzyme that depends on vitamin B12 as a cofactor. And so it's already been pretty well established that levels of MMA increase in people who have vitamin B12 deficiency, and also vitamin B12 is known to decrease during age. However, they also show that that isn't likely to be the full scenario. And so another interesting finding from this study is the fact that MMA is more effective at causing this pro-tumorigenic response at lower concentrations if MMA is in a lipidic structure, because this aids transport of MMA inside the cell. So what is MMA doing to the cell once it's inside? Well, this isn't still fully understood, but what they observed was an upregulation of a transcription factor known as SOX4, and SOX4 is already a marker of poor prognosis that contributes to tumour progression in a variety of aggressive cancers. And interestingly, SOX4 depletion fully abrogated the ability of MMA to promote migratory and invasive properties of the cells. But it seems likely that MMA is impacting the TGF-beta signaling pathway that is upstream of SOX4 to achieve this impact. So I'm sure further studies will elucidate this a little bit further, but it highlights the importance of metabolic reprogramming to the relationship between aging and cancer. So going on from lipidic structures of MMA now to functionalized lipid-like nanoparticles for in vivo mRNA delivery and base editing. So what's this all about? Well, many diseases occur due to a deficiency of a protein. For example, hemophilia A can occur due to a lack of human factor VIII. And so in terms of treating these different diseases, it's not trivial just to be able to give back a protein, neither is it trivial to be able to edit the source of the genetic mutation in the first place. And part of this comes down to the fact of the difficulty of getting the materials to the right place at the right time. And so what is explored in this paper is the advancement made in the development of lipid-based nanoparticles that can carry mRNA. And so mRNA is the transcript produced from DNA, which is the intermediate between DNA and protein production. And so if a cell receives mRNA, it can then convert it into protein and bypass the DNA stage. And so in the study, they screened a variety of different functionalized lipid-like nanoparticles, and they found that one of these compounds, which they referred to as FTT5, was able to efficiently deliver long mRNAs, in this case, human factor VIII, for the treatment of hemophilia A. And so this was done in mice. And so what they found was that the delivery of this protein, which enables blood to clot to treat the symptoms of hemophilia, was able to produce enough of this factor within 12 hours to reach 90% of what is seen in normal activity. So as well as this actually being effective, it was also shown that there was no apparent organ damage within the mice as well. But the more interesting example that they used was a lipid-like nanoparticle that contained the components of a base editor. So they had the mRNA encoding the base editor and also a single guide RNA to target the base editor to the desirable sites on the DNA. And so this is different. So this time we're not just producing a protein, we're producing a protein, which is the base editor and the machinery to be able to actually edit DNA. So it's kind of combining two treatments in one. And this was pretty cool. And they show a high percentage of base editing when they try to target the gene PCSK9. The protein encoded by this gene is important in cholesterol clearance, and PCSK9 is commonly mutated. And so these sets of experiments are pretty exciting in terms of potential therapies in the future. And notably, the technology has been licensed for further clinical development. So lastly, today in this video, we're going to look at genetically modified mosquitoes that have been okayed for a first US test flight. And so mosquitoes can carry a variety of different diseases, such as dengue. And the most common used technique to eliminate outbreaks or bursts of mosquitoes is by using pesticides. And so not only are pesticides somewhat ineffective, they also have adverse consequences for other wildlife and for the environment. But an alternative strategy is to reduce the rate of reproduction of these mosquitoes. And one way that you can do that is through genetic modification. And so there's an English biotech company known as Oxitec that have developed normal-looking male mosquitoes that actually contain a gene that they call a self-limiting gene. That means that when they mate with a female, any of the female offspring die whilst only the males can survive. And so obviously you need the female to reproduce to create more offspring. And so if you've only got the males surviving, you effectively shrink the population size of these swarms of mosquitoes. And the technology that actually enables this is pretty neat. So this self-limiting gene that they refer to is basically a gene that gets expressed within only the females. And when it gets expressed, it promotes its own expression. And so you have tons of this one protein within the cell. And this protein actually binds to the transcriptional machinery that basically prevents other genes from being expressed. And so without all these other essential genes being expressed, the female larvae actually die before becoming adults. And so obviously one concern with genetically modified organisms is always the safety and whether or not the technology can spread to other organisms. But the nature of this genetically modified system is such that it is self-limiting. And so it means that it can't establish in the ecosystem and it would only target their own species. And moreover, it is environmentally sustainable since it prevents the use of chemicals. So I guess if all goes to plan, they will be released in 2021 in Florida Keys. So that brings us to the end of these brief news articles. As I said, this is the first time I've done this. I don't know if this is good, bad, or whatever, but I find it interesting to do. So that's all I've got to say. So I hope you've learned something and thanks for listening.